Now this is the last video on chapter 6 and this is the component 6.1 part 4. It says describe the semi-conservative replication of DNA during the S phase of the cell cycle. Now if you remember the cell cycle, this is the part which is the interphase, all this part which is the 3/4 of this whole circle, and this would be G1 and this would be G2, and this of course is the part which is the most important part, and this is the S phase of interphase. So it is called the S phase of interphase. This is of course from the previous chapter. So this is the S phase of interphase, and during this phase DNA replication takes place. That means the chromosome number, which is 46 in humans, will become 92 chromatids. So DNA replication is going to occur in the S phase. This is called the S phase. S stands for synthesis phase. So the S phase of interphase, this whole part, this whole thing here, this up to here, is all called interphase. So this is the part where the DNA replication, and this DNA replication has been given a special name, which is called semi-conservative, and semi, you know, means half. So when the original strand of DNA, when it's copied and two copies are made, and each copy will have one of the original and one new, so there's one old and one new. That's why it's called semi-conservative. So semi-conservative replication is going to take place in the S phase of interphase. Now the DNA replication, the first step is that the DNA replication, it's going to unzip the double helix. So I mean, here you can see it's still zipped up, but here I've shown you that it is unzipped. So it unzips, and there's an enzyme called helicase which is going to break the hydrogen bonds, because if you remember, there's going to be A here, so it's got to be T here, and this was joined together by the hydrogen bonds. So the hydrogen bonds are going to break, and this is going to, the complementary bases which were held together will no longer be held together, and now they are these two separate strands. So helicase, then the separation of the two single strands of DNA creates a Y-shaped. This is that Y-shaped structure which is called the replication fork. So this would be the replication fork which has been formed, and now the two strands will act as a template. Template means whatever the bases are present on this strand, and whatever the bases are present on this strand, now these are going to dictate which other bases are going to come and get attached to it. So now one of the strands, you know, is oriented in the 3-5 direction towards the replication fork. Now this is called the leading strand. And the other is called the lagging strand. So this one which I'm drawing here in blue is going to be called the lagging strand. So we have the leading strand and the lagging strand. Now what we have to understand is that the leading strand is going to be copied in a continuous manner. So this is the leading strand. Now that is going to be copied continuously. So it's a continuous process, and this would be the 5 end and this would be the 3 end. And this of course will continue like this. Of course it's not going to continue, it's going to continue. So this is going to be the leading strand which is copied, and this is going to be a continuous copying. But we have a problem with the lagging strand. The lagging strand is copied discontinuously. So it's going to be copied in little bits of, and these fragments have a special name, and these are called Okazaki fragments. and these are called Okazaki fragments, these little pieces of DNA. And then of course we have an enzyme which is going to join them. The lagging strand, because it's sort of chunks of DNA which are called the Okazaki fragments, so this is called a discontinuous. But this of course is later on joined, and there will be an enzyme which is going to join these gaps, and we're going to have a continuous, and that enzyme is called ligase, which is going to join it together. So I'm just showing the joins like this slightly. Now this is going to be joined by the enzyme DNA ligase, which is going to join these Okazaki fragments together. So now the DNA strand is now copied, so half of it is the original, which as you can see we've shown in the black, and the other part is in red. So the original strand was all in black, and now we have, so please understand this is that the semi-conservative replication means that it's going to be half old and half new. In semi-conservative replication, what you have to understand, if the original DNA had heavy nitrogen, so 2 out of 2 are heavy nitrogen. But if we give it light nitrogen, and I color that in red, so in the first division we're going to have 2 out of 4. Now when this is going to divide again, now how is this going to change? Well, this one goes here, this one goes here, and this goes here, and this goes here. Now when new DNA is added to it, I'm just going to make it a little squiggly line. So we have this, we have this, we have this, and we have this. So now this is going to be 2 out of 8. Now I can exactly remember an MCQ which just tests whether you've understood this or not. So the original strand were 2 out of 2, then it's 2 out of 4, then it's going to be 2 out of 8, and then it's going to be 2 out of 16. So this 2 is going to remain as many times as it divides, but the 2 original ones will always remain. So this is one of the MCQs which checks whether you have understood this part of the semi-conservative replication or not. And that should be the end of chapter 6. And I hope you've understood this chapter. And this is better now, and you can do all the MCQs on it.